Friday, everyone. Welcome to the first of our series of Timothy A. Johnson Medical Scholar Seminars. We have about 8 throughout the year, and it's a seminar series that the School of Medicine sponsors, really with the idea of promoting the importance of interaction between our physician researchers and our full-time scientists who are working on biomedical research, as well as across the continuum of epidemiology, public health. We feel that it's really important for both our TBMH graduate students and our medical students to see exemplars of people who have made a career out of practicing medicine and conducting impactful research. And so I'm very excited about our first speaker, and I'm equally excited to be able to introduce our third-year medical student who nominated Dr. Fairchild. So you're going to hear an introduction from Mia Edelson for our first speaker. Mia.
Good afternoon, everyone. Thank you for being here today, and a huge thank you to Dr. Karen Fairchild for taking the time to participate in the Timothy Johnson Medical Scholar Lecture Series today. My name is Mia Edelson. For those of you who don't know me, I'm a third-year medical student here at the Virginia Tech Fralin School of Medicine, and I'm delighted today to have the opportunity to introduce Dr. Fairchild, who I was lucky to be able to work with at the University of Virginia as a clinical research coordinator. Dr. Fairchild is the interim chief of neonatology at the University of Virginia. She has led several NIH-funded studies, has over 120 publications, and her works have been cited over 6,500 times. She has several research interests, including using vital signs to predict neonatal outcomes such as sepsis and necrotizing enterocolitis, ultimately leading to early interventions to reduce morbidity and mortality in this especially vulnerable population. Dr. Fairchild graduated from Wellesley College. She then took several gap years, which we'll talk about, and went on to attend Duke University for medical school. She completed her residency in pediatrics at Johns Hopkins University and her fellowship in neonatology at the Children's Hospital of Philadelphia. She then went on to join the UVA Department of Pediatrics in 2004. Dr. Fairchild has been a mentor to me for several years, and what initially actually connected the two of us is the fact that we both majored in French in college and went on to pursue our respective careers in medicine. Working for Dr. Fairchild, I witnessed firsthand how she manages to balance providing exceptional care for her patients and performing innovative and groundbreaking research. For all the med students in the room, I know you recently started about 6 weeks ago, and I'm certain that Dr. Fairchild will teach you some helpful lessons today as we all embark on our respective careers in medicine. So without further ado, let's give a warm welcome and round of applause to Dr. Fairchild.
Thank you so much, Leslie and Mia. It's really a pleasure to be here. I added a subtitle to my title, so 10 lessons from a very long career. I met some of you at lunch today. I started medical school in 1985, so it's been an extremely long career. And the thing I really most want to impart to you is that I still love my job at my age, and I hope that most or all of you will be able to say that when you're my age. All right, so first of all, I don't call myself a physician scientist. I feel a little bit like an imposter when I say the word scientist. So I'm, definitely a researcher. And I also, in my research, collaborate with other clinicians like nurses, nurse practitioners, therapists. So that's why I call myself a clinician researcher. So this is me at age 5, playing doctor. My little sister is the patient. So, I always wanted to be a doctor. Nobody could quite tell why. My parents were academics, not medical. And so they weren't sure why. I think I was born wanting to be a doctor. But I wasn't sure I could do research. I really, like Mia said, I was a French major in college. I was very insecure about the whole science thing. And in fact, when I was trying to decide where to go to medical school, I'm from North Carolina, so it was between UNC and Duke. And Duke requires a solid year of research. And in 1985, it had to be a solid year bench research. And I was totally intimidated by that. But I went to Duke and I actually did molecular immunology research and really liked it and found out that I could do it. Maybe. I wasn't sure I wanted to do it for a career still. And it really wasn't until more than a decade later that I'm like, oh yeah, I do want to do this for a career. So you're going to notice some water images in a bunch of my slides. This is a painting by Roland Anderson that was hanging outside of the UVA NICU for many years. And we finally made them take it down because we said, it looks like this woman is struggling in the water. You know, she kind of looks like she's swimming, but she doesn't look too comfortable. And this kind of describes me in a lot of my career. I felt a little bit like I was working hard. I was swimming, but I wasn't sure that I wasn't going to drown. But I didn't drown. And so one important message, lesson number one, is don't be afraid of deep water. You have to jump into the deep end of the pool to learn how to swim. And that will help you get places. All right, lesson number two, non-traditional paths and gap years can be good. So like Mia said, she was a French major in college. I was a French major in college, very non-traditional. And also Mia took some gap years, I took some gap years. So some people are what we used to call straight arrows. I don't know if you call it that anymore, but you know, high school, college, graduate school, postdoc, career. And that's fine. It's a great way to go for some people. I took 2 gap years between college and medical school, worked in a lab in Bethesda, and again, actually liked the research and found that I maybe could do research in that gap year. And the second gap year I took was between residency and fellowship. I didn't decide on neonatology until my 3rd year of residency. So it's fine if you need to take a year off. I worked in an emergency room between residency and fellowship for a year, and those were good years. So I've also taken some turns in my research and gotten onto different research paths. So it's fine to do that as well. And actually my career looks kind of like this line, some left turns, some right turns. But notice that all of these arrows get to the same place. All the arrowheads end at the same place. So just might take you a little bit longer to get there. And the important thing also, if you decide to take gap years, is it's better to call it a bridge year. So a bridge is something used to get someplace else. A gap sounds like something you fall into. So think about what you want to do during your gap years. This is an internet image that I found saying things to do in your gap year that look good on your resume. So kayaking for your gap year for the whole year is not going to look good on our resumes in this room. But it is okay to do some fun things during your gap year, but it's better to do something that's more like a bridge. So this is the Golden Gate Bridge. It's a way to get from one place to another. Apparently people actually swim from one end of the Golden Gate Bridge to the other. Sounds totally crazy to me. That would not be me. But yeah, bridges can be better than gaps. All right, you all know who Mia Edelson is now. Some of you knew Mia before, and now you know what she majored in French, or in college, French. So non-traditional path, and as Mia told you, she also worked for 2 years as a research coordinator in neonatology at UVA. So that was a chance for her to get a sense for what it was like to do research, what it was like to be in the NICU and the delivery room. So I think those were good years and helped propel her to medical school. All right, lesson number 3, seek mentors. And the important letter in that word is the S at the end, so you need multiple mentors. You all have probably heard this 1,000 times, but it does ring true. And so I just wanted to show you some of my mentors. If you start at the upper left, my mom and dad, like I said, they were not medical doctors, but they were both Dr. Fairchild. So they were both academics. And I think part of the reason that I'm standing in front of you today and have had an academic career is because I think it was in my genes, in my environment. I didn't really realize until I was an adult that maybe my parents actually had some impact on my career path. But yeah, mom and dad, and then there's like, I don't know, 12 or more other people there. Over the course of many years, I've had many great mentors, and you will too. It's important to seek them out. Don't wait for somebody to be assigned to you as a mentor. You really need to find somebody who's a good fit for you. And then the other thing is that even at my age, I have mentors. So I'm doing research. I still feel insecure about a lot of things. So I reach out to people who know more than me. And, you know, nowadays I call them colleagues more than mentors, but I still do have mentors, and some of my mentors are actually younger than me. All right, so the next lesson, number 4, it's fine to take time off. I almost changed the word fine to great or important here because time off is key. I had 2 maternity leaves early in my first faculty position when I was at University of Maryland. And, you know, back then in the 90s, it was kind of 6 to 8 weeks of maternity leave. And nowadays you get more and you should take more. And dads should take time off too when they have a baby. So this is an emperor penguin family. If any of you know anything about these penguins, the dad participates a lot. So the mom lays the egg and the dad actually incubates the egg for, I don't know, weeks or months or something, while the mom goes and forages for food. And then when the egg hatches, mom comes back and they switch roles and dad goes out and forages for food. So it's important to take time off when you have kids. It's important to take time off if your family needs you for whatever reason. And also take care of yourself, because you have long careers ahead of you. And if you burn out, that's a problem. So if you have either physical health issues, mental health issues, stress, anxiety, depression, take time off to get better. Okay. When I was a first-year fellow at CHOP, very intense time, a lot of sleep deprivation. I got very depressed in my first year of fellowship, and I didn't tell anybody. I was kind of embarrassed to tell people at work, to tell my friends, to tell my family. So I struggled for a while, and I ended up not having to take time off work, but I ended up finally being able to reach out and get help. And after that, I got better. So nowadays, there's a lot more appreciation for the importance of the commonness of mental health problems. And so there's also more acceptance of telling people that you need time off or you need treatment. And so take care of yourselves, take care of your health. All right, everybody knows the Titanic, right? And everybody knows that there are icebergs out there and your ship can sink if you hit an iceberg. So I put this slide in here because lesson number 5 is don't be afraid to jump ship if you feel like you're going to hit an iceberg, right? It's better to get on a different ship than to go down with the ship. So this lesson I learned when I was at University of Maryland, first faculty position. I had been in her lab for 2 years studying FC receptors in gram-negative sepsis. My mouse model wasn't working out. My FC receptor antibodies weren't working. Nothing was working. I took 2 maternity leaves. I felt like my ship was going to sink. And I actually thought maybe I should just get out of research. Maybe I should work part-time. My kids were little. Maybe I should not go full force with career. Fortunately, somebody actually came along and recognized that my ship was about to hit an iceberg. And that person's name, he's a physician scientist, Jeff Hasday. He recognized, because he knew my lab mentor, and he actually was the husband of one of my neonatology colleagues. And so Jeff came to me and said, Karen, this is not working out with FC receptors. Why don't you come into my lab? He was studying fever effects on inflammatory cytokines. And I said, well, fever is not really relevant to babies. What about hypothermia, which is really relevant to babies? And he said, yeah, sure, study hypothermia. You can use all of the resources that I have in my lab. I will mentor you. I promise you will have a first author paper in 2 years. And I said, Jeff, you can't promise that. You don't know how the data are going to look. He's like, don't worry, we'll work it out. So in 1998, I switched labs and I switched foci, whatever, and actually started doing hypothermia research. So just to tell you about phase 1 of my hypothermia research. So how many of you have heard that hypothermia is neuroprotective? Yeah, for both adults and babies. So if you cool the body down to 33 degrees Celsius, the injured brain will do better. So for babies with birth asphyxia, hypoxic ischemic encephalopathy, we cool them now to 33 degrees for 3 days and they have better outcomes. And the mechanism of hypothermic protection is multiple. It's not a silver bullet, it's like a machine gun. It probably has 20 different mechanisms. One of them was thought to be anti-inflammatory, but we started studying hypothermia in monocytes, so in vitro, and found increases in pro-inflammatory cytokines, tumor necrosis factor alpha, interleukin-1 beta. And that kind of was counterintuitive. So we thought maybe hypothermia is a two-edged sword. It's neuroprotective, but maybe it's also ramping up pro-inflammatory cytokines, which can be neurodetrimental. So maybe there's something here that we could study. So we published a couple of papers. Like Jeff said, within 2 years, I had a first author paper. And this was looking at both febrile range temperatures, because Jeff and his lab were interested in fever, and clinically relevant hypothermia. And we looked at it just at the very most basic level. We took cultured human monocytes from newborns and from adults, and we showed increased pro-inflammatory cytokines with hypothermia and with hyperthermia. And then we looked at the mechanism. And so at least part of the mechanism is through hypothermia activating NF-kappa B, which is a transcription factor that drives pro-inflammatory cytokine gene expression. So that was exciting. Lesson number 6. And how many times have people told me this? You have to have protected time to be successful in research. And this is the tip of the iceberg slide, but I'm using it to sort of represent the right balance, 80-20, 20% clinical, 80% research, especially if you're in the lab doing bench research, because all that time that I was in the NICU, I was not running gels, doing Western blots, doing immunohistochemistry, developing, in vivo models and that kind of thing. So I really, at Maryland, didn't have protected research time, even though I was starting to get some traction with my research. I had a couple of publications. I got an American Heart Association beginning grant in aid, so it wasn't huge. But still, my division chief and department chair said, sorry, we can't give you less clinical time. We need you in the NICU. So 20 weeks in the NICU was kind of not tenable for developing an in vivo model to test out what I was finding in vitro. So lesson number 7, moving institutions can be a good career move. It was hard to leave Maryland. My kids were little, so they didn't care so much, but my husband, he had a job in Annapolis and he was like, well, I guess we could move. But he, we kind of moved to Baltimore for his career as a magazine publisher. And he said, well, I'm turning 50. I guess now would be as good a time as any. The kids are little. We could move. if you find a good place. And University of Virginia just so happened to invite me to give grand rounds. And then they said, oh, by the way, we have a faculty position. What would it take to bring you here? And I said, protected research time. And this was in 2004. And Bob Chevalier was the chair. And he is a physician scientist. He's now an emeritus professor, but he did amazing bench research and clinical research. And he gave me a 3-year startup package. So I went to UVA, had protected time, a lab, money for research tech, got all set up, and went into phase 2 of hypothermia research, which was trying to translate in vitro to in vivo. So in the first couple of years, it took a while to get my mouse model set up. So I published a couple of papers based on work that I did at Maryland. So the first paper was a DNA array, a microarray study looking at gene profiles at 33 in THP1 cells. It's a human monocyte cell line, but putting the cells at 33 degrees or 37 degrees and looking at gene expression profiles. So that was one of my Maryland papers that was published after I moved to UVA. And then we also looked a little bit deeper at the mechanism of NF-kappa B activation. So we saw that at hypothermic temperatures, you can imagine that a lot of enzyme kinetics are different at 33 degrees compared to 37. So we found that 33 degrees led to enhanced phosphorylation of I-kappa B kinase. And so NF-kappa B spent more time in the nucleus of the cells, driving gene expression. So those 2 papers happened in the beginning of my time at UVA. And those 2 papers helped me get a K08. Have you all heard what a K08 or a K23 is? Some people are saying yes or no. So it's a mentored clinician scientist development award. K08 is for bench, and K23, which I'll reference later, is for clinical research. So my K08, I got it 8 years out of fellowship, which is almost unheard of. I call it my late K08 because, and the only reason NICHD gave me this grant is because I moved to UVA and was working with Marty Mayo, who's an NF-kappa B expert. He was assistant professor when I first went to UVA and then was promoted to associate. So it took 2 submissions of the K08 because at first they said your primary mentor is too junior. And then I wrote him a P&T letter and Marty got promoted to associate. So on the second try, I got the K08 in 2006. And that gave me 80% protected research time to spend in the lab. So the next lesson is lesson number 8, independence is overrated. So, you know, a K08 is a mentored grant, but everybody says, oh, then you have to make the K to R transition. And an R is a quote unquote independent award. But, you know, there is no independence in the word research, right? I mean, in science, there's an I for independent, but if you're doing NIH funded research, you have to collaborate. So independence is impossible, I would say, in biomedical research, NIH funded research. So MDs need to work with PhDs and vice versa. This is Jeff Hasday, MD in the white coat, Ishwar Singh, PhD. And they were my 2 mentors in my last 6 years at Maryland who helped me get the hypothermia work going. You know, Jeff is a pulmonologist and he's had multiple R01s, but he's taking care of patients some of the time. Ishwar, you know, runs the show in the lab and he has some independent funding as well, but the 2 of them completely relied on each other for the collaboration to be successful. This is Marty Mayo, who was my K08 mentor, NF-kappa B expert, and me in the white coat. By the way, I never wear a white coat except for photo ops like this. but Marty was really instrumental in getting me the K08. This is Joel Linden, PhD, and Bob Streeter, MD, in the white coat. Joel Linden is a phenomenal scientist, studies adenosine receptors. And in his lab, he had a lot of PhDs, some MD PhDs, and then he invited me to be part of his lab because I was studying hypothermia impact on lung inflammation. He had a program project grant on lung inflammation. So I kind of went to lab meetings and was developing, helping to develop mouse models to study how hypothermia might have anti-inflammatory effects in the lungs. And, you know, at Joel's lab meetings once a week, I vividly remember a graduate student presenting her mouse model of ARDS, acute respiratory distress syndrome, and she flashed up a histology slide, and Bob Streeter and I looked at each other and were like, that doesn't look like ARDS, because, you know, we had studied histology and ARDS, and it's like, we're not sure that that's a good model. So she went and tweaked the model and then showed another slide, and we're like, yeah, that's ARDS. So, you know, I think it helps for MDs and PhDs to work with each other. There can be some animosity, and, you know, Leslie and I are talking about this, and I've been in labs with PhDs where they were making fun of the MDs not being true scientists, and I've been in labs with MDs where they're like, well, the PhDs don't understand what's clinically relevant. I'm like, we all have to work together if you're going to benefit patients, if you're going to get NIH funding. Yeah, so that's lesson 8. All right, lesson 9, don't get sucked into the clinical vortex. I actually did get sucked in. I'm still here, so I came out of it, but, you know, as a clinician, sometimes the path of least resistance is to go take care of patients, because like I felt, you know, I love taking care of babies. The NICU was right there in the next building over from my lab. And sometimes I felt like, yeah, I think I'm going to just go do some patient care for a while. And the more you do that, the less chance you have of being successful in the lab. So the way I really got sucked into the clinical vortex is that the fellowship program director at UVA left when I was in my second year of my K08. And I actually had a couple of fellows doing mouse research in the lab with me. And Phil said, Karen, you should be program director. And I said, Phil, I can't. And he said, well, nobody else can either. So I kind of got sucked into that. And I love education. I spent 12 years as the fellowship program director, but it took a lot of time away from the research. So I'm not making excuses, but I'm just kind of warning you that you will be asked to do more and more clinical things, or administrative things, or be on committees. And you have to learn how to say no, especially early in your career. All right. Lesson 10, follow the data. The data don't lie, right? Sometimes the data don't match your hypothesis, but you have to follow the data anyway. So here's the painful lesson that I learned, is that what I found in monocytes didn't translate to mice. And, you know, my whole K08 was based on preliminary data that was in vitro, and aim 1 of the grant was develop a mouse model. And so I developed the mouse model and figured out how to make mice hypothermic to 33 degrees, which is how cool we make the babies for neuroprotection. And I actually didn't find that TNF and IL-1 were increased. What I found was that IL-10 was increased. And IL-10 is an anti-inflammatory cytokine. So I kind of found the opposite thing to what my hypothesis was. So we published this, and then I had a dilemma. You know, do I do aims 2 and 3 of my K08? And to me, that made no sense at all, because I disproved the hypothesis that that hypothermia in vivo was going to be pro-inflammatory. It really seemed like it was anti-inflammatory. And aims 2 and 3 were figure out the mechanism by which hypothermia is pro-inflammatory in vivo. So it didn't make sense to do aims 2 and 3. So, my second choice was find the mechanism of increased IL-10. And I did that a little bit in vitro, but then I realized it was sort of reverse translation. We were already cooling babies. We knew that hypothermia was neuroprotective. So did it really matter why IL-10 went up? Not to me. That was not a path I wanted to take. Number 3 path was jump to a different line of research in the mouse model. And NIH gives you money, it's venture capital. You don't have to do aims 2 and 3 of your KO8. You have to submit an annual progress report and nobody reads it, I'm pretty sure, except somebody in administration. So I wrote a progress report after the end of my second year and I'm like, the hypothesis was wrong and I'm not doing aims 2 and 3. Here's what I'm doing. And I said, I'm pursuing the mechanism of increased IL-10 in vitro. And then, the next year I'm like, why am I doing this? NIH doesn't care. They just want me to do something good with money, right? They don't want me to pursue a dead end. They want me to pursue something that's going to work out. So I should say that at this point, my other choice was just give up research altogether because, you kind of come to this turning point and you're like, is this really worth it? But I don't know why, I just love research. So I kept going. So here's what I did. I switched gears in 2008 to study how sepsis alters heart rate characteristics. So this sounds like 180 degrees from hypothermia, which it is, but let me explain why I thought this was a good question. So here's my line, right? I took turns and here I am. So I did arrive somewhere. So here's the story. Babies in the NICU, and that's a preemie, are connected to leads. And so we have a monitor and we're monitoring their heart rate, the electrocardiogram signal, right? And so at UVA, before I even got there, Randall Morman and Doug Lake and others developed the HERO monitor. And so what the HERO monitor does is it takes a clinical observation to a computer algorithm to a score. So it basically, the observation was that babies, when they're in the preclinical phase of sepsis, so they're not crashing, they're not obviously septic, have decreased heart rate variability and transient decelerations of their heart rate. Okay, so Randall and Doug and a company in Charlottesville turned that into an algorithm that gives you a score called the HERO score that is the fold increased risk that in the next 24 hours, the baby is going to crash with sepsis or a sepsis-like illness. I've given so many talks on the HERO monitor and the autonomic nervous system. I could talk forever, but I won't. So this is just a quick example of normal heart rate characteristics on the top. That's 30 minutes of heart rate, and you can see little accelerations and decelerations. And then on the bottom, obviously abnormal heart rate characteristics. So no variability and superimposed decelerations. And so if you were an obstetrician and you saw that heart rate pattern, you would say emergency C-section, crash C-section, because that's a fetus that's in trouble. And it's often hypoxia, ischemia in the fetus, but it could be chorioamnionitis and a systemic inflammatory response in the fetus. It turns out in babies in the NICU, the #1 cause of that tracing is sepsis. So what I did, because I had this mouse model where I surgically implanted a little telemetry device that measured temperature for my hypothermia work. And I'm like, I spent all this money and all this time developing this model. Oh, these telemetry devices have little ECG leads. So I could actually monitor the electrocardiogram in the mice And then I could make them septic, something I can't do in the babies. In the babies, you have to wait for them to become septic. In the mice, you can make them septic. So the model actually worked out pretty well. And we discovered 2 mechanisms of abnormal heart rate patterns in sepsis. Number 1, cytokines. So the model was E. coli, LPS, or TNF. The finding was decreased variability, and that was published in 2009. And then I'm really summarizing a few years of work in one slide. But we also found that when we gave the mice gram-negative, gram-positive, or fungal pathogens, whether intraperitoneal or intratracheal, they had transient decelerations in their heart rate, just like the septic babies have, or the asphyxiated fetus has. So, and it was modulated by the vagus nerve. So that was fascinating to me. So I tried to get an R01. So here's my attempt to make the K08 to R01 transition with bench research. So you see at the bottom, so eventually when you all are writing NIH grants, you'll be able to go into the system and see what your history is. And so my history on the bottom there is K08 in green. I don't expect you to be able to read any of this, by the way, but then the very next one up, the title is cholinergic activation and heart rate characteristics in sepsis. Triage. Rejected. NIH doesn't write rejected, they write administratively withdrawn, but that's a nice word for rejection. And then I submitted 3 other grants that got rejected on the clinical side, but there is a happy ending to the story. So in 2010, I moved from the bench to the bedside. I kind of thought mouse research was hard. It turns out research on little 500-gram preterm babies is pretty hard too, but it's fun and rewarding. So that's why I'm still doing it. And I got lucky ×3 because I eventually got 3 grants. So one of them is related to the sepsis early warning system. I'll tell you a little more about that. One of them is related to apnea of prematurity, and one of them is related to delayed cord clamping and helping preemies breathe before you clamp the cord to reduce the risk for intraventricular hemorrhage, brain hemorrhage, which is an unfortunate, not uncommon complication of prematurity. All right, so the sepsis early warning systems work involves looking not just at heart rate characteristics, but we're adding oxygenation patterns to the algorithm to try to develop an even better score than the HERO score. And we're looking at biomarkers and physiomarkers for sepsis and also necrotizing enterocolitis. That's the alphabet soup of prematurity, right? So we have NEC, IVH, intraventricular hemorrhage, BPD is bronchopulmonary dysplasia. We've actually discovered heart rate characteristics predicting cerebral palsy in NICU patients. And, CP isn't usually diagnosed until beyond 6 months or even beyond 12 months. And autism, we published a paper with a developmental pediatrics fellow showing that spikes in the heart rate during the neonatal period predict increased risk for autism diagnosis down the road for boys, but not girls. So that was interesting. Yeah, so again, independence is overrated. This is a multi-PI grant. Randall Moorman and I are the PIs on this. We're currently in our 9th year, I guess. This is if you go on NIH Reporter, you can pull up the information about current and past funded awards. All right. Then there's apnea, central apnea of prematurity. All preemies less than 28 weeks and most preemies between 28 and 32 weeks have central apnea of prematurity. They grow out of it. This graph on the upper left, so UVA developed an apnea detection system that involves analyzing the chest impedance waveform data and looks for periods where there's a flat chest impedance signal together with a dip in the heart rate and a dip in the oxygen saturations. So we call these ABDs. And that heat map kind of shows you gestational age on the y-axis and chronologic age on the x-axis. And you can see that babies of lower gestational age, lower chronologic age have more ABDs, and then they outgrow it. But we actually got a UO1, which is kind of like multiple RO1s. And this was with 5 other NICUs to study apnea of prematurity. And again, multi-PI, this time Doug Lake and Randall Moorman are the PIs on the Prevent Leadership and Data Coordinating Center at UVA, and I'm a co-I on that grant. All right, and this is my last grant that I'm going to show you about. This is Vent First that Mia helped with. So it's a 12-center randomized clinical trial of assisting ventilation during 2 minutes of delayed cord clamping for babies born at less than 29 weeks gestational age, with the primary outcome being intraventricular hemorrhage. And this again is multi-PI. John Catwinkle and I are co-PIs on this grant. We just ended enrollment in Vent First this past spring. And so now we're in data cleaning and data analysis mode, and then writing the paper and presenting it in December. Oh, wait a minute. Let me go back. That thing on the top, there is a baby in that photograph. So this is, we did a pilot study, and our fellow had a GoPro camera on his head. So we took some photos and videos, but basically Vent First involves, a lot of these babies are born by C-section. Okay, so in this photo, you can see the OB on the right, you can see his hands on the umbilical cord, and you can see the incision. And then you see 2 neonatology providers. You basically just see their gloves. And there is a baby, and the fellow is giving the baby positive pressure ventilation, because these babies often just are apneic when they're born. So you can't delay cord clamping for 2 minutes if the baby's not breathing. So Vent First was a lot of work. As Mia can tell you also, you had to get consent from these moms who had preterm labor and were threatening to deliver a baby at less than 29 weeks, and they're on magnesium, and which makes you feel terrible. And, you know, getting consent from these moms was challenging. All right, so just a couple of suggestions about how to get lucky in research. And I put lucky in quotation marks. Of course, it takes hard work. And it also takes some creativity and flexibility. So one message is ride the waves at NIH. You know, hypothermia in 2005 was a very hot topic, because all of these RCTs were coming out showing neurologic benefit for babies if you cooled them, but people didn't know the mechanism. And my in vitro work showed the opposite, like maybe hypothermia is pro-inflammatory, and maybe hypothermia plus some kind of anti-inflammatory drug might even work better for these babies. So that was one wave that I rode. Apnea of prematurity, also NHLBI was kind of interested in apnea of prematurity when we submitted the Prevent grant. And delayed cord clamping is a hot topic in neonatal resuscitation. And John Catwinkle is a very famous neonatologist, so he was kind of the obvious person to take that on. Meet experts in your field and at NIH. I used to think it was mostly about the science and working hard and publishing a bunch of papers and writing a really great grant. It turns, and I'm an introvert. I might not seem like it because I'm giving this talk, but I'm basically like happy to not talk to anybody all day and just look at data. But I learned kind of the hard way that you actually have to talk to people, meet people in your field. So don't just go to a meeting and look at everybody's research and talk about your own research. Go to a meeting and find out who the experts are going to be at that meeting. Email them ahead of time and say, hey, I'm really interested in your paper. Could we meet for coffee or something. And also meet people at NIH. So you have to call your program officer if your grant gets triaged and find out, you know, what their suggestions are about how to improve the chances. So build collaborations, and I put the word strategic in here. I have a lot of collaborators and most of them have been productive, but I have been asked to collaborate and I've also collaborated in things that were not very productive. And so you kind of have to pick and choose. And so if somebody asks you to collaborate, you know, if you're young, you're going to be like, yeah, sure, I'll collaborate. But you need to pick and choose carefully, because if you end up going in one direction with a collaborator that's maybe not a productive direction, like you might get one paper, but it's probably not going to get beyond that, you might want to focus your efforts toward what you think might be a more productive collaboration. All right, getting close to wrapping it up. I finished my 10 lessons, but I had to put in a bonus lesson. Pay it forward. This is Bryn Sullivan, currently assistant professor, but about to be promoted to associate professor. Bryn was a pediatrics resident at UVA, came to me and said, I want to do research. Her dad is MD, PhD, you know, so it was in her genes too. But I said, oh, there's this hero monitor project that I've been wanting to do that's feasible in the context of being a resident. So she did that project with me, got a first author paper, did her fellowship at UVA, got a couple more first author papers on vital sign analytics, and then joined the faculty. And she was in the first class of I Thrive Scholars. So that's a collaboration, as you know, that includes Virginia Tech. And so Bryn got an I Thrive, which is what I call a pre-K award, but basically gave her 2 years of protected research time while she wrote her K-23. And then she got her K-23, which is on integrated physiomarker, biomarker, and clinical predictive analytics for early warning of sepsis and necrotizing enterocolitis in very low birth weight infants. I told her that the title was too long, but she put it there anyway, and she got the grant. So, so she got the K-23, and then the really happy thing is that she just recently was awarded an R-18. I didn't even know what the R-18 mechanism was. It turns out it's a 3-year grant, and it's basically the same amount of money as a 5-year R01, but compressed into 3 years. So she had submitted a couple of R01 applications. One got triaged, one got scored, but not great. And this one ended up getting funded, this R18. It's not her favorite area of research. In fact, she has done no research in neonatal abstinence. But basically, she's the expert on vital sign analytics, and this is a collaboration with WashU St. Louis and University of Alabama, Birmingham, so 3 NICUs. And what they're doing is they're taking the work that Bryn has done in her K-23 on sepsis prediction using pulse oximetry data, so heart rate and SpO2 data, and using that to predict need for narcotic therapy for babies who are having withdrawal, if that makes sense. So she's, she calls it POWS for NOWS. NOWS stands for neonatal opioid withdrawal syndrome, and POWS is a pulse oximetry warning score. So she basically, NIH really is interested in quantitative measures of neonatal withdrawal, since a lot of women are on either prescribed or non-prescribed substances and the babies have withdrawal. So deciding which baby needs to stay in the hospital, which baby can go home, which baby is going to need morphine tomorrow, that kind of thing. So my next to final slide, again, the water theme. So this is a painting called Swim Team by Rebecca Kincaid, a Vermont artist. And this is just at the exit of the elevators on the 7th floor of UVA Hospital, which we call UVA Children's. UVA Children's is just one floor of the UVA Hospital. We're trying to grow, but we're meeting resistance. anyway, but I love this painting and I see it every day. And it reminds me, first of all, you need to be a good team member. And second of all, you need to enjoy the process. So these kids are taking a break from swimming and they're enjoying each other. So it's really important not just to focus on the prize at the end of your career, but to actually enjoy all the steps along the way. And then finally, acknowledgements. So the UVA Center for Advanced Medical Analytics, CAMA, Randall Mormon, Doug Lake, and Katie Cron have been really instrumental in my research. Obviously, thank the funding sources, including startup money from University of Maryland, startup money from UVA. And of course, I have to thank my neonatology colleagues because, again, it takes a team. I am in the NICU 14 weeks a year. Some of them are in the NICU 20 weeks a year, and they're also doing education and administration and a lot of other things. So it's important for the clinicians and the researchers in our division to get along and appreciate each other's contributions. And actually we added 3 more. So this is an old slide. So we have 3 that we added last summer. All right, and that is my final slide. So I would be happy to take questions.
That's true. Yeah. Hi. I had a question. I mean, we get pretty lucky here with, early exposure. I'm a med student. We get pretty lucky here with early exposure to our PhD colleagues, but I was wondering if you had any advice on kind of like fostering those connections as you get into residency and career and stuff too, since you said it was so critical.
Yeah, I would say happy hours. I think if you get to know your colleagues outside of work, it can help a lot with reducing some of the work-related tensions. I have set up 2 happy hours in the next 2 weeks with different research collaborators and clinical team. And, you don't necessarily have to drink, but if you want to, that's fine, or eat, actually eating together. I think just getting to know people as people, and then trying to, and I've been in a lot of environments where there was tension between the MD and PhD researchers. And I think it's hard. I think PhD researchers have a lot harder job because you have to generate your funding. And as a neonatologist, I could spend 5 minutes taking care of a not very sick baby on CPAP and generate a lot of revenue for the hospital so that when I need protected time, they leave me alone. If I were a developmental pediatrician or an endocrinologist or something else trying to do research, it would be harder because I wouldn't be generating all those clinical dollars for the hospital. But I would say, I've sort of grown up. My mom's PhD, my dad had a DVM and a master's, but he did biomedical research. And so I kind of grew up with parents who weren't MD researchers. So I appreciated, especially once I got to be an adult, I certainly didn't appreciate their research when I was a kid. But, I appreciate that you have to collaborate. And especially if you're doing health-related research, it's not straight science. So you need to get along. Happy hours might help. Oh.
Love it.
Yeah, sorry. Glad meeting you, Dr. Fairchild, and it was an amazing presentation. You've made mention about the fact that collaboration is very important, especially in work settings, right? what about having experiences outside work settings? Because of course, the toll that a career could put on you could be very high. And is it important, or since you've had like a very long career, do you have like a safe space outside of your career where you could just be yourself, where you could just be the little girl that you showed us up in the first slide,
Yeah.
and how relevant is it for you to have that whilst sustaining your career? Is it very relevant?
Extremely relevant. Safe space. Yeah, my safe space is playing my cello. I didn't do it so much when my kids were little. Actually, I didn't do it at all. I had a long gap where I didn't have that music outlet. But then I got it back again, and I played in the Waynesboro Symphony until COVID hit. So music is part of my safe space. I also like watching baseball games with my husband. So right now we're watching the Yankees be awful. But yeah, you definitely need to have time away from work. Those 2 gap years that I took, some of the best years of my life, because I had more free time and I could really reflect on what do I really want to do with my life. And, I think sleeping is important. And eating is important, and exercising is important, and then having some passions outside of work. So do all of those things, and to do all of those things and have a successful career. I just feel really fortunate that I was able to. And, I think you have to keep trying. Yeah. And I also think, getting back to the question of the relations between PhDs and MD researchers, is so I like to invite people to come to the NICU and see the clinical relevance of what they're working on. And then I actually love to go to people's labs. And I really enjoyed yesterday and today, hearing about people's research here. And I think that all of you are really fortunate to be in this medical school and have exposure to research throughout your 4 years. I only had that 1 year at Duke, and honestly, they crammed all the basic science into the first year. It was a nightmare. It really was. And then I had a research year, which was at the Pasteur Institute in Paris. So that was a lot of fun, but I did a lot of lab work, but with the Eiffel Tower in the background. But then, the last 2 years at Duke were really hard. And so I think, and I did no research those last 2 years. So to me, it makes sense to kind of spread it out. I also think this is an advantage over UVA. And I didn't wear any orange, and I'm not a football fan, whatever. So, I think you all have an advantage over UVA medical students. I work with them sometimes in research 7 weeks in the summer after their first year. That's not enough time. And then in their 4th year, sometimes they come back to me and say, could I do a little more research to get authorship on that paper that we talked about? So they do a little bit first, at the end of first year in the summer, and a little bit in their 4th year. I was at University of Maryland, and all the peds residents were required to do research. And so because it was a requirement, they did it, and they collaborated, and then we made it fun, and some of them got publications. They all had to present their research. So I think requiring research is important, and making it fun. Any other questions? We don't have the iPad power here, guys. We do have grown-ups now. I know, and I think this is a beautiful place to live. It really is. Thank you so much.